Thank you for volunteering your time to help monitor the coastal water of Elbow Key.
Before we begin the technical training, let us begin with a little background on what we are measuring and why it's important.
Water is a central component of all life on Earth. As an island nation, the Bahamas are intricately tied to water.
The health and well-being of the water relates directly to the health and well-being of the country. The Bahamian economy relies highly on tourism, and the tourists come to spend their time near the pristine turquoise waters. There are many things we can measure that reflect the cleanliness of water. The indicators that are used will depend on what a given water source is used for. In this program, we will measure a parameter called fecal coliform. Fecal coliform is a collection of bacteria that grow in the gut of all warm-blooded animals, so potential sources of fecal coliform include birds, pets, livestock, and humans. By measuring fecal coliform, we are estimating the contamination load of feces in the water. This is an important parameter to measure in water that humans will come in contact with because feces can contain many infectious agents that can make people sick. We use fecal coliform to alert us of the presence of feces in water and thus the potential threat of disease. As you may notice, birds, pets, and livestock do not use toilets, so their feces often make their way into water. This is not always problematic. What matters is the quantity.
The more feces, the more likely that there will be infectious diseases in the water.
Because of this, there are standards that the United States Environmental Protection Agency recommends for what levels of fecal coliform are tolerable for given water uses. This is why we will be measuring quantity of fecal coliform in the waters around Elbow Key.
The purpose of this program is to collect a long-term data set of fecal coliform levels at different locations in the waters around Elbow Key. This will help us get a better picture of the overall health of the waters. This data set will be a tool to direct further efforts to improve water quality.
Before we begin the details, let's walk through the highlights of what water quality monitoring entails.
Although each volunteer will be responsible for a part of the process, it is important for you to understand the process from start to finish. The first step is preparation. Before collecting the samples, the bottles must be cleaned using a 5-step process. Using the clean bottles, samples are collected at a couple of locations around the island. Samples should be dropped off at the laboratory as soon as possible, but must be at the lab within 3 hours of being collected. At the laboratory, the samples are filtered to remove the bacteria from the water. The bacteria gets trapped on filter paper, which is then put in a petri dish with food for the bacteria and placed in the incubator overnight. This creates the perfect environment for the bacteria to grow. Each bacteria that was in the sample grows into a bacterial colony composed of many bacteria, which is visible as a spot on the filter paper. These spots can be counted the next day. In this way, we are able to tell how many fecal coliform bacteria were in the sample, giving us a concentration of fecal coliform. Now we will walk you through the entire process step by step. You can watch each piece on its own, or you can watch the whole process in its entirety. Ensuring the cleanliness of your bottles is essential to collecting good samples. Achieving this cleanliness requires a 5-step process. To prepare, set up a basin of soapy water, a basin with cistern water, and a Brita filter with reverse osmosis water.
You will want to be near a sink and have your rubbing alcohol on hand. Begin by washing the bottles in the soapy water. Make sure to clean the cap and the threads of the bottle as well.
Next, rinse in your cistern water basin to remove suds. Following this, fill a bottle quarter full with water from the sink, shake and empty, making sure again to get the cap and threads. Repeat using reverse osmosis water. And finally, rubbing alcohol. Always make sure to clean your cap and threads as well. Dry off the outside of the bottle using a clean towel so that tape will stick. Apply fresh labeling tape so that you know the bottle is clean.
The bottles are now ready for collecting samples.
Before beginning, each sampler should make sure that they have all the materials they will need for the sampling trip.
These include a cooler filled with ice, sampling bottles with blank labeling tape on them, a permanent marker, data sheets, a clipboard, a thermometer, a bucket, and rope.
When you arrive at the sampling site, make sure that it is safe and appropriate for you to take a sample. Label the bottle you are going to use with the date, time, sample location name, and ID, and your initials. Make sure to write on the bottle before it gets wet. Next, you want to begin filling out the water quality data sheets. Measure the temperature of the water using the thermometer.
First, triple rinse the bucket. Draw a bucket full of water and dump away from where you are sampling. Here we are using a bucket to collect the sample. Even if you do not need a bucket, the same principles will apply. Avoid stirring up the water where you will be pulling your sample from. Avoid dunking your hands in the water and empty the rinses away from the location you are sampling. Fill and dump the bucket 3 times, then draw a final bucket full. When you are ready to draw your sample, you want to be careful not to contaminate your sample. You should have clean hands, but don't use an alcohol-based hand sanitizer, as this can interfere with the sample as well. From the bucket, triple rinse your sampling bottle. Keep your hands away from the mouth of the bottle and try to keep them out of the water as much as possible. Be sure to dump outside the bucket and rinse the cap and threads each time. Fill and dump 3 times. Then draw your sample, filling the bottle all the way. Recap and place in the cooler.
Return the sample to the laboratory as soon as possible, but no later than 3 hours after collecting the sample.
Before leaving the site, make sure you have filled out your data sheet.
Analysis of samples will take place in the laboratory. First, plug in and turn on the incubator. Make sure the temperature is set to 44.5 degrees Celsius. It will take a little while to heat up. While the incubator heats up, gather all the necessary equipment. This includes sample bottles, permanent markers, petri dishes,
media. These are little plastic ampules with purple liquid,
filters, a box of gloves, plastic pipettes for dilutions, filtration units, vacuum pumps, rubbing alcohol, tweezers, a lighter for flaming the tweezers, reverse osmosis water, and a Brita filter. To begin, put on rubber gloves. In this video, rubber gloves will not be used, but you should use them whenever they are available. Your goal throughout this process is to avoid contamination of the samples. Remember that the table you are working on is not sterile. The running of every sample, whether it is the sample water, a negative control, a lab blank, or a positive control, should be done in exactly the same way every time.
Take a new petri dish and label with a permanent marker. You should write the date the sample was taken, the site ID, your initials, and the volume of sample filtered. Label both the top and bottom the same way.
Place the Petri dish on the table with the side that says millipore face down. Take one media ampule and remove the cap. Using your other hand, remove the cover of the Petri dish. Be careful in doing this that you handle it as little as possible. Hold the cover by the top edge only. Do not place it on the table, as the table is not sterile.
Empty the entire contents of the media ampule over the pad in the Petri dish. Replace the cover of the Petri dish and set aside for now.
Next, sterilize your tweezers by dipping the tips in isopropyl alcohol and holding them to a flame. Make sure they are dry before touching anything with them so that you do not kill off bacteria in your sample.
First, make sure your filtration unit is sterile. Sterilize the filtration unit with a small amount of alcohol swished around through both chambers of the unit. Rinse the unit twice with reverse osmosis water filtered through a Brita. Whenever you are pouring water out of the pitcher, be careful not to let the lip of the pitcher touch the other vessel.
Shake sample bottle to mix the water, then prime the filtration unit with a small amount of sample water.
Now we will set up the filtration unit. Remove the lid. Then remove the top half of the filtration unit and place carefully on the lid. Do not place it directly on the table, as the table is not sterile.
Take a filter and open the packaging, being sure only to touch the outside of the package. Inside the package there are two things: the white filter itself and a blue sheet that is there to protect the filter. Using your sterilized tweezers, separate the blue piece from the white filter. Grab the filter carefully with the tips of the tweezers. Remove from the packaging and set the packaging aside, as the inside is sterile and will be of use to you in storing your tweezers later. Center the filter on the base of the filtration unit with the grid side up. The little bit of water on the top is okay. In fact, this will help the filter stick in place while you replace the top of the unit. After you replace the top of the unit, make sure you press down firmly so that you have a solid seal. Attach the pump to the side spout of the filtration unit. Shake your closed sample bottle again and pour 100 milliliters into the top of the filtration unit. Get your head at the level of the filtration unit so you can measure accurately. Now you will begin filtering the sample. Slowly and carefully pull the plunger out. This may be a little difficult at first. If you have someone else nearby, it may be helpful to have them hold the filtration unit steady. You do not want to pull too quickly. Watch to make sure you have a slow and steady drip of water through the filter. As you do this, any bacteria that is in the water will be deposited onto the filter paper.
Any debris in the water will also become stuck on the filter paper. The dirtier a sample you have, the more difficult the filtration may be. But do not worry about debris in your water.
Continue pulling the plunger out, but be careful not to pull it all the way out of the pump. If you need to reset the plunger, carefully remove the pump from the spout and slowly push the plunger back in. It is a good idea to keep your hand in front of the attachment nozzle, as there is a little piece of cotton that sometimes comes shooting out if you push the plunger in too quickly. If this piece comes out, simply put it back in the nozzle and use a small blunt object to push it back down into place. If you use your tweezers, be sure to re-sterilize them. Replace the pump and continue filtering until all the water has been pulled through the filter. If you are filtering 200 milliliters of sample in total, carefully remove the hand pump and pour the water from the bottom chamber out in the sink. Replace the pump and repeat the process of adding 100 milliliters and filtering it. When you have filtered the full quantity, remove the upper chamber and place it on the lid. Remove the vacuum pump from the filtration unit. This will make it easier to remove the filter. Resterilize your tweezers using alcohol and then a flame. Again, make sure they are completely dry before proceeding. Now you will remove the filter paper from the filtration unit. This can be tricky. It may take you a couple of times to do it just right, but the more you practice, the easier it will become. Be very careful not to tear the filter as you do this. It is quite fragile. Try to touch the filter as little as possible. Try to touch only the very edge, not the middle. A good trick that you might find works best is to run the tip of the tweezers around the edge of the filter, barely even trying to get under it. Now that you have the filter, hold it firmly with your tweezers in one hand. Remove the cover of your prepared Petri dish with the other hand. Again, do not put the cover on the table and try to touch only the edges of it. Place the filter paper, grid side up, in the Petri dish, trying to center it as best you can. It may be easiest to lay the filter on one of the edges of the Petri dish and then slide the filter over until it drops into the plate. Replace the cover of the Petri dish tightly and flip the Petri dish over so that the side saying millipore is up now. Keeping the millipore side up, place the Petri dish or plate in the incubator. That is the process for filtering samples. Next, we will talk about how to analyze the plate for colonies. After the plates have incubated for 24 hours at 44.5 degrees Celsius, they are ready to be counted. At this point, any fecal coliform bacteria that were in the water were trapped on the filter paper. Then, when we put the filter paper in the plate with the media, they were encouraged to grow under optimal conditions. Any bacteria that could possibly have survived to infect a human will have grown into big colonies of bacteria that can be seen with the naked eye. Each colony is made up of identical offspring from a single bacteria. When counting bacteria colonies, you want to look for dark, blue, well-delineated circular spots on the filter paper. Here is an example of a plate with 3 perfect colonies. Sometimes you may have no colonies. This is actually what you hope for, especially on your blank plates. Sometimes you may have colonies that are different colors. Colonies that are different colors, such as gray or cream, are not fecal coliform bacteria. These colonies will not be counted. When you only have a few colonies, counting is simple. When you have a large number, it is important to keep track of your counting. This is why there is a grid on the filter. Use the grid lines to keep track of which colonies you have already counted. Count all the colonies in one row, including the colonies that lie on the line below that row. In the next row down, you will count all the colonies in the row and those that lie on the line below, but not those lying on the line above, because you have already counted those. It is a good idea to have more than one person count each plate. If there are discrepancies, take the average of what each person counts. Thank you so much for volunteering your time and energy into this program. This is a big step for the future of the waters of Elbow Key.